Hello, my name is Lisbeth Tejera and we'll be talking about the classification of point mutations. We're going to talk about a couple ones including transition, transversion, silent, missense, nonsense, non-stop, and stop gain mutations. We will start by defining what it means by point mutations. A point mutation, also called a singular nucleotide polymorphism or SNP, is a simple change in one single base pair in the DNA sequence. That's it, just one nucleotide out of thousands that we have within our system. Even though it's such a small change, it can have very different outcomes. Sometimes it changes the way protein is able to work, sometimes it affects the gene regulation, and sometimes it has no effect at all. We usually classify point mutation in two different ways. First, by the type of base substitution it is, whether it's a switch between similar bases or completely different ones. And second, by the effect on the protein, for example, whether it changes the amino acid sequence or stops protein production early. Okay, now let's break down the two main types of base substitution. We have transition and transversion, as you can see by the image. A transition is when one base is replaced by another of the same type. That means when a purine like adenine or guanine is replaced with another purine, or when a primine like cytosine or thymine is replaced with another purine. However, transitions are known to be more common because the chemical change is relatively small. The structure of the base doesn't change as dramatically as a transversion. A transversion is when a purine is replaced with a primine, or vice versa, when a primine is replaced with a purine. So an adenine or a guanine is meant to be changed to either a cytosine or a thymine, or the other way around. These are known to be less frequent because the substitution involves a more significant structural change, and the key point here is that transition and transversion are defined solely by the type of base substitution and not by the effect on the protein. That comes later when we classify the functional impact of the mutation. Now that we classify point mutation by the type of base substitution, let's look at the functional consequences on the protein. A silent mutation changes the DNA codon but not the amino acid it codes for. Because the protein sequence doesn't change, these mutations typically have no effect on protein function. Then we have a missense mutation, which changes the codon so that it codes for a different amino acids. Depending on the location and the properties of this new amino acid, this can have little impact or can significantly change the protein structure and function. Then there's also a nonsense mutation, which converts a normal codon into a stop codon. This leads to premature termination of translation, producing a protein that's often non-functional. Then there's also non-stop mutation, which does the exact opposite. The normal stop codon is lost, so the translation continues beyond its usual endpoint, leading to an abnormally long protein. Finally, the term stop gain mutation is essentially another name for a nonsense mutation because the new non-stop codon is gained where one shouldn't be, again resulting in early termination. To clarify a little bit what I just went over, here we have this image that shows us a little bit of a clearer picture. So first we're going to start off with silent mutation, which again changes the DNA codon, but the new codon still codes for the same amino acid. So here in our original DNA, we have UUA, which corresponds to the amino acid of leucine. And then for silent mutation, now we're changing one U to a C, encoding for, instead of UUA, it's going to code for CUA, but still the original DNA and this new mutation, they're still coding for leucine. So next we're going to go into the missense mutation, which remember, changes the codon so it codes for a different amino acid. So when our original UUA is coding for leucine, the mutation one, which is going to be GUA, is going to be coding for valine. So as you can see, the amino acids for the original DNA and the new DNA, it's different, leading to the missense mutation. And this substitution can potentially alter the protein structure and function depending on what type of amino acid that it's changing to. And finally, there's non-stop mutation. which can also be known as a stop gain mutation, which changes codon for the amino acid into a stop codon. So as you can see, the UUA, which is leucine, now is being coded as UAA, which is a stop codon. This prematurely stops the translation and usually produces a shortened and non-functional protein. Now that we have a better understanding of point mutations, what types there are, and their consequences, we're going to go into a little question. So we have a patient that's having his gene sequence. The test reveals that he has a single base substitution changing UAU to UAA. Which of the following best describes the mutation? We have A, silent, B, nonsense, C, transition, D, missense. I'm going to give you a couple of seconds so you can answer. Okay, hopefully you answered by now. As all of you may know, the answer is B, nonsense. So the codon now codes for a stop signal, which is that UAA, leading to a premature termination and a possibly non-functional protein.